So I guess my journey to becoming a virologist was fairly standard. As a high school student, I was really fascinated by infectious diseases, and so I enrolled in a undergraduate degree program in microbiology. And it was really doing that program, and then I also performed an internship at Inhibition Biosciences, where I worked in the virology department. And then my first full-time job was at the Institute of Cancer Research, working with Robin Weiss on HIV. And I think just throughout that whole time, I really became fascinated with viruses, and particularly their amazing complexity and diversity. And that's really stayed with me throughout my whole career. And even now, I find it difficult to focus on a single virus and become engrossed by any new virus that comes along. I think that's really why I started working in emerging viruses in particular. So to move on to the talk today, I'm going to talk about some protective human monoclonal antibodies that we've developed against Chikungunya virus. And then I'm also going to present a little bit of data that we've gathered from the ongoing Chikungunya outbreak in the Caribbean that Michael mentioned earlier. So Chikungunya itself is derived from a Makonde word describing the physical symptoms of the disease, which is really characterized by an acute onset of fever, and in particular, a severe pain in multiple joints. The disease itself has a fairly low mortality rate, less than 1 in 1,000 cases, but we're really interested in this virus because it causes persistent and relapsing joint pain that can be both severe and very chronic, lasting over 2 years in some cases. So Chikungunya is an alpha virus. It's, as with all alpha viruses, it's mosquito-borne, predominantly by Aedes aegypti and albopictus. There's 3 genotypes, with the Asian strain being the current strain driving the outbreak in the Caribbean. Alpha viruses are really characterized particularly by the fact that the E1, E2 membrane glycoproteins form this very structured and highly organized icosahedral glycoprotein shell that surrounds the viral membrane. And in particular, I'm going to keep coming back to this, and this highly organized structure is key to many aspects of the virus life cycle, from driving viral assembly and budding, all the way through entry, where conformational changes in the glycoprotein drive membrane fusion, and then finally through to uncoating of the viral genome. So up until a couple of years ago, Chikungunya was mainly confined to the tropical and subtropical regions of Africa and Asia, and really fairly closely matched the geographical distribution of the mosquito vectors. However, in December 2013, the virus was first detected in local transmission in the Caribbean, in particular the French territory of St. Martin. However, it rapidly spread throughout the Caribbean and then to Central America, and is now north in Mexico and south in Brazil in reasonably high numbers. And currently the CDC estimate that there have been nearly 1.5 million suspected cases. However, I'm going to present some data that suggests that this may be a gross underestimate of the actual number of infections. And whether Chikungunya can establish itself in the US is open to question. There's certainly been a lot of traveler-introduced cases, and in Florida there's been at least 1 instance of a localized transmission in about 12 individuals. And the mosquito vectors are present, particularly Aedes aegypti and albopictus in the southeast corner of the US. So there are the conditions available for the virus to establish. The CDC predict that there aren't going to be major outbreaks in the US, based mainly on air conditioning, that people just aren't exposed to mosquitoes the same way they are in other countries. So I'm just going to present some of our data we've gathered recently from Puerto Rican blood donors during this recent epidemic in 2014. So this study was funded by the CDC, performed by ourselves at Blood Systems, in collaboration with our testing arm, CTS, who collected the samples from blood donors in Puerto Rico, and then our commercial partners, Hologic, who provide many of the nucleic acid testing assays for routine blood screening. So the study was divided into two arms, a sero-survey looking at pre- and post-epidemic, and then RNA testing performed by Hologic with an experimental Chikungunya dengue duplex assay on their Panther platform. And this is performed throughout the 2014 epidemic. Firstly on mini pools that for routine blood screening, donor samples are pooled into mini pools of 16 prior to routine HIV, etc., nucleic acid testing. And then they also performed about 3,000 tests on individual donation from the very peak of the epidemic. So looking at the serology first, we used a commercial IgG ELISA for chikungunya, and we detected no confirmed positives in the pre-epidemic sample set, which was collected as soon as we got word that there was localized transmission in Puerto Rico in June and early July. And then we also tested about 1,000 samples from the post-epidemic in March 2015. And you can see that a large percentage of the blood donors are strongly positive for chikungunya IgG. There was a few intermediate samples. One was confirmed by IgM testing, and the other three were indeterminate by Western blot. So they may have been very early stage immune responses to an acute infection, or more likely it's probably cross-reactivity with other alpha viruses, which is pretty common for chikungunya, for alpha virus serology. But just concentrating on these positives and the one IgM confirmation, we found 23.5% of these post-epidemic blood donors had seroconverted just in the 9-month period of the epidemic in 2014. And if this is extrapolated to the whole population of Puerto Rico, which is about 3.5 million, we would see over 800,000 infections just in a 9-month period in one island in the Caribbean. The CDC reported that there was about 35,000 suspected cases in Puerto Rico. So there's a couple of explanations for this apparent discord. We've had many anecdotal reports that chikungunya is so prevalent in the Caribbean now that most people that are infected are simply just not bothering to go to their doctor because they know that there's really nothing specific to treat the virus. And however, there's also likely a pretty high percentage of asymptomatic or subclinical infections. And this is a point of contention that for the African strains, it's been thought that over 80% show symptoms, but some reports suggest that the Asian strain, which is causing this outbreak, may have lower level of symptomatic. Moving on to the nucleic acid testing, Hologic found that in our mini pools, they were finding at the peak of the epidemic in September and October, over 15% of the mini pools were reactive for chikungunya RNA. And then looking at the individual reactives, about 1,000 each month, it peaked at just over 2% in October. So 2% of blood donors donating in these Puerto Rican studies were RNA positive for chikungunya. However, so obviously at BSRI, we're interested in this for the potential of blood transfusion transmission of CHIK. However, there actually have been no confirmed cases of chikungunya transfusion, despite the very high viral loads that the virus can reach in the plasma. There has been one confirmed case of the related alpha virus, Ross River virus, being transmitted by red blood cell transfusions. And it may just be that the background level in endemic areas is so high, and obviously you also have recipients who are already immune to the virus that you really just miss these transfusion cases. So we're going to be following this up with more dedicated recipient-donor linked studies in the future to really see if we can identify transmission of chikungunya. But we did want to see if at least these highly positive individual plasma samples, if given as a mock plasma transfusion to mice, would transmit the virus. So Hologic also provided us with estimated viral loads for these samples, and we put a range of these different plasmas into a mouse model of chikungunya. In this case, it's an interferon receptor knockout mouse, so obviously it's immunocompromised compared to many real blood transfusions. And we put in about the equivalent of half a unit of plasma. And you can see that there's a pretty good correlation with time to death in these mice and the viral load that we put into them, even down to samples which were at the limit of quantifiability were still able to transmit virus, and we were able to isolate the virus from these mice. So it's not necessarily proof that CHIK can be transfusion transmitted, but it's certainly suggestive that it may be. So the main subject of my talk is going to be on these human anti-CHIK monoclonal antibodies that we've developed. So there's currently no licensed vaccine for chikungunya, and there are no specific therapeutics. Monoclonal antibody therapy has been suggested by a number of people, and this could be useful both prophylactically, used to treat at-risk individuals such as pregnant women and the elderly, and also, more importantly, used therapeutically, both to alleviate acute symptoms, but also to reduce the more serious chronic disease that follows in many individuals. And these chronic symptoms have been linked to the presence of persistent viral RNA and presumably also viral replication. And it's been shown in mouse models that robust adaptive immune response can alleviate these chronic symptoms. So we used memory B-cell cloning to identify a number of human monoclonal antibodies, primarily from a small outbreak that occurred in Italy. And then our long-term collaborators, Integral Molecular, used phage display to identify additional antibodies from the Reunion Island outbreak in 2006. So we found many neutralizing antibodies, a lot of non-neutralizing antibodies too. I'm just going to concentrate initially on a couple of them, C9 and CKV63. They both inhibited live chikungunya virus replication very potently, and they were relatively specific in that we didn't see inhibition of a range of related alpha virus envelopes. The antibodies were also potent in a couple of in vivo models of chikungunya. Firstly, in collaboration with Andreas Serha in Australia, we tested them in a mouse model of acute arthritis. So these mice develop joint swelling in their feet about 7 to 10 days after Challenger virus. And prophylactic treatment with relatively low doses of our antibodies are able to completely alleviate the foot pad swelling. We also tested them in a therapeutic model. In this instance, this is neonate mice, 8 to 9 day old, and they die relatively rapidly after Challenger virus, between 4 and 6 days. And we are now able to go out to about 36 hours post-infection with our antibodies at relatively low doses and get at least partial protection from death. So one of the issues with monoclonal antibody therapy is that the virus can rapidly evolve escape mutants. And so we looked at this by making escapes to our two best antibodies, both in vitro and in vivo, and using different strains of the virus. And we found in each case, after sequencing the full structural gene of the virus, that there was a single point mutation giving escape for each antibody. For C9, we found glycine at position 95 in E2 was mutated to either E or arginine, and this gave, in both cases, almost complete resistance to the C9 antibody, while still being sensitive to 63. When we made escapes to CKV63, again, it was a single point mutation at this tryptophan at position 64 in the E2 protein, either to glycine or arginine. Again, the virus, particularly for W64G and to a lesser extent W64R, was resistant to CHIKV63. However, in this instance, it was also resistant to our other antibody, C9. So we suspected that they had overlapping epitopes. And this is pretty much confirmed by a collaboration with Yifan Cheng at UCSF, where we performed cryo-EM analysis of, in this case, our C9, where we made an FAB fragment and bound that to virus-like particles of chikungunya. And you can see in the structure that both the G95 specific selection from C9 and the W64, which was selected by the CHIKV63 antibody, both fall within the epitope footprint of the FAB. We also performed some competition analysis, particularly focusing on the W64 mutation, as it gave escape to both of the antibodies. And for the W64R, it could outcompete wild-type virus within 3 passages in culture. In contrast, the W64G mutation, even after 3 passages, maintained about the same level as the initial input, suggesting it couldn't outcompete wild-type, but equally wasn't outcompeted by the wild-type. And we saw a similar pattern in vivo, firstly in an acute model, where after 4 days post-infection, we harvest tissue and then sequence the full structural gene. And we saw that whatever ratio of virus of W64G to wild type we put in was basically maintained after 4 days in the acute model. We also looked in a chronic model. So this is looking at the RNA persistence, which is linked to chronic disease. And after 28 days, we harvested both the left and the right ankle tissue from the mouse. And really, regardless of what ratio of virus we put in, we saw about an equal level of wild type and W64G in the mouse, again suggesting that there was a lack of competition between the 2 viruses. However, despite this, when we look at pathogenicity, so this is in the neonate mouse model again, where the mice die 6 to 9 days after infection. We saw death with both the wild-type virus and the W64 mutation, but only 1 mouse died throughout the whole course of the experiment with W64G, suggesting that escape from our antibodies may be driving attenuation of the virus and a loss of pathogenicity. We obviously have to repeat these experiments in other models and particularly look at whether there's any attenuation of chronic disease or not. So moving on to mechanisms of action. Firstly, we performed some time of addition studies with our antibodies. So if we pre-bind antibody and virus and then add them to cells, we see all of our neutralizing antibodies neutralize infection. If we add virus to cells first and then add our antibody, we find loss of neutralization by this antibody E2, which is a commercial antibody that maps to the receptor binding domain of the virus. So it's probably not surprising that it appears to be blocking a attachment stage of infection. And then finally, we performed fusion from without or acid bypass assays, where we, again, bind the virus to the cell and then add our antibody and then give a low pH pulse, which should trigger conformational change in the envelope glycoproteins and mediate membrane fusion at the cell surface. And again, our 2 main antibodies, 63 and C9, completely blocked fusion from without, suggesting that they're inhibiting at a fairly late stage of entry, probably within the acidified endosome, and preventing conformational changes. So that's all well and good. There are 2 well-known mechanisms by which antibodies inhibit many different viruses. However, Jing Jin, a postdoc in my lab, very perceptively noticed that when she left antibodies in the culture, they inhibited much more efficiently, over 10-fold more efficiently than if the antibodies were washed out. And she performed a number of experiments doing washout studies, time of addition studies, a single round of replication, and using inhibitors to inhibit additional rounds of replication, and really found that the point at which many of these antibodies were blocking was when the virus was being released. And you can see here where you bind the virus to the cell surface. You then wash the virus for 3 hours to allow entry to occur, then wash the virus out, then allow antibodies. We see very potent inhibition of both viral RNA and viral infectivity released into the supernatant. In fact, even more potent than entry neutralization. And this is most dramatically demonstrated here using some antibodies from Mike Diamond. These are mouse antibodies. And unlike many of our antibodies, they're very poor on non-neutralizers. However, they still inhibit viral budding very potently. Mike Diamond has gone on and challenged animals with many of these antibodies to give protection, and found that despite the lack of neutralizing activity, they are relatively potent at protecting against infection. Our escape mutants pretty much mirrored the entry neutralization that we saw escape for both viruses with the W64G, both antibodies with the W64G mutant. Jing then went on to look at mechanism. She made FOB versions of our antibody and found that while FOBs were slightly less potent for entry neutralization, there was a complete loss of budding inhibition with these FOBs, suggesting that we require cross-linking by a MAB of the viral glycoproteins on the cell surface in order to mediate membrane budding inhibition. So for a couple of viruses, there have been budding inhibition or viral release antibodies described, particularly Marburg virus. However, when they performed EMs, they found that basically the antibody was cross-linking glycoprotein on the virus and the cell, and just tethering the virus to the cell. However, when we performed a transmission EM in collaboration with Cynthia Goldsmith at CDC and Larry Ackerman at UCSF, in no antibody or control antibody experiments, you can see alpha viruses, fairly typical budding from the plasma membrane. However, when we add our inhibitory antibodies, we see no evidence of viral budding and no virus particles in the extracellular milieu. Instead, what we see is these viral cores all accumulated just below the plasma membrane. And as shown in more detail here with immunogold labeling, that we see the plasma membrane surface decorated with our antibody, with the immunogold label and our antibody in the deembolic glycoprotein, and the viral cores clustered below the surface. We also performed electron tomography on embedded samples in collaboration with University of Colorado Boulder. And this is the control cell, and again you can see viral particles budding at the cell surface. However, when we added our antibody, again, you can see there's no evidence of viral particles outside the cell. Instead, we're seeing again the immunogold labeling of our antibody and glycoprotein complexes at the plasma membrane, and all these viral capsids all accumulated just below the PM. So the current hypothesis for how alpha viruses bud is that assembly is primarily driven by the glycoprotein shell formation, and in particular, lateral interactions between the glycoproteins. And as shown in more detail in this paper from Holland Cheng a couple of years ago, that these hexagonal array of viral glycoproteins are delivered to the plasma membrane from internal vesicles. And they're arrayed in hexagons, but they quickly undergo conformational rearrangement to introduce pentagons, which are required to form a sphere, just like a soccer ball. And it's this rearrangement that really drives membrane curvature and formation of the viral particle. So our hypothesis is that budding inhibition mediated by our antibodies occurs by cross-linking of E2 in these highly ordered membrane lattices, and it prevents this lateral rearrangement of the glycoprotein tertiary structure into these pentagons, and therefore prevents this membrane curvature formation. So we're currently collaborating with Misha Sherman and Scott Weaver at UTMB, who are fortunate enough to have a cryo-EM facility in a BSL-3 lab to really see if we can look at the atomic level by cryo-EM to really see the formation of particles in inhibition by our antibody. So I think I've mentioned most of the people throughout the talk, but I'd just like to acknowledge Jing Jin in my lab, who really drove the second half of this talk, as well as Nathan Liss and Kai Lu, and then Marcus from many of the animal experiments. And our collaboration with Mike Diamond at University of Washington, where we're trying to characterize some of these antibodies. So I'll stop there. Yep.
Do the mutations, the escape mutants, have any phenotype in mosquito? Have you tested that?
So we've done some preliminary growth kinetics in mosquito cells, but no actual mosquito studies yet. But the G95 mutation is slightly retarded in terms of growth in C636 cells. And that position actually matches a known mutation in Symbis virus that is known to have lower growth kinetics in Aedes aegypti midgut. So there is the possibility that the escape does interfere with mosquito transmission, but yeah, we haven't done those studies yet. Yeah.
So am I understanding correctly with the last part of the talk? Are you assuming, is that, is the idea that some of the antibody is coming into the cell and preventing that release? How do you?
No, the binding. So as soon as these glycoprotein lattices get to the surface, then they're binding at the surface and just then preventing the subsequent conformational changes.
Can you show us where the antibody bonds on the structures there?
I can't really show it on this structure. Basically, these are the E1, E2 spikes kind of sticking out here, and it's binding in this cleft between two of the monomers within the heterotrimer. So kind of these trimeric spikes here, so kind of down in the middle of these trimers.
Do you think those have more potential in terms of neutralizing, viral infection in general than, just binding to the virus? I mean, just binding the fusion one, the one that are involved in fusion, which is a really tiny moment in the viral life cycles, which is those that's probably much longer, and So do you think there's more?
Yeah, I mean, I think separating some of the different actions is important, and so we're doing some of those experiments with Mike Diamond now. So he certainly has non-neutralizing antibodies that still inhibit, and some potent neutralizing antibodies that don't work in vivo. So, there's an issue with alpha virus, for certainly other viruses, I think that'll be the case, but with alpha viruses, because they have so many glycoproteins on the surface that you basically have to coat the whole surface with the antibody to really be effective. So.
I was wondering, I remembered one of your slides, you talked about serological, or you could only see RNA evidence of the virus and without serological specificity. Did I get that right? And so I'm wondering, maybe you explained this, but I didn't quite capture just how that process works. That is, you have abundant evidence, apparently, nucleotide-based, evidence of the virus, and yet, you're getting no antibody action.
Do you mean in the Puerto Rican study?
Yeah, or I guess my specific question is, with immunohistochemistry, for example, could you identify viral presence in a preparation?
So in chronic samples, yeah. So there have been some evidence of viral antigen as well as viral RNA persisting, but I don't think really that's been worked out particularly well particularly in humans, in some animal models, you can isolate infectious virus out of these persistent samples. Yeah.